Good day to one and all present here. I am SITRARASI from Madrasa Women's University, Kodaikanal. Today I will be presenting about the organisms that are commonly used in the process of bioremediation. First, I will like to give a few introduction about the topic. According to the Environmental Protection Agency, bioremediation is a treatment that uses naturally occurring organisms to break down harmful contaminants into less toxic or non-toxic compounds. This is the definition given by EPA. It can be categorized into several forms depending on the organisms which are utilized for bioremediation. Among these organisms, plants and microorganisms are the most used, and they play a significant role in the process. First, I would like to talk about phytoremediation. In 1991, the term phytoremediation was coined. This is a plant-based remediation technique which has lots of promise when it comes to accumulating and immobilizing persistent pollutants. Plants are natural filters that digest chemicals that are created by the environment. Phytoremediation is a new technique that is used for removing pollutants from soil and water using plants. So, phytoremediation procedures are normally divided into 5 categories. This categorization is based on the contaminant's fate or destiny. So, naturally combinations of these techniques are found as well. So, the first procedure is phytoextraction. It is also called as phytoaccumulation. It is a process in which plants accumulate pollutants in their roots and in their shoots. This method is very cost effective. So, second process is phytotransformation. This is also known as phytodegradation. It is a process that involves the uptake of organic pollutants from the soil or sediments and water, and they are transformed into a less hazardous or transportable substance. Metal chromium can be taken as an example. It can be reduced from hexavalent to trivalent chromium, which is less mobile, and it is a non-carcinogenic form. So, third one is phytostabilization. This technique involves reducing the contaminated soil's mobility and migration. This is done by adsorption. Leachable elements bond to the plant structure and they form a stable mass. This prevents the toxins from re-entering into the environment. So, the fourth type is phytodegradation. This is a process in which the pollutants are broken down by the activities of the rhizosphere in the roots of the plants. Then the presence of proteins and enzymes produced by the plants and soil organisms like bacteria, fungi also helps in the process. The final process is rhizodegradation. This is a symbiotic interaction between the plants and the bacteria that have evolved over time, which help in the breakdown of pollutants. Final one, okay, I would like to talk about MYCO-REMEDIATION. Now, for the breakdown of contaminants or waste, many fungi, including mushrooms, include enzymatic machinery. As a result, their potential can be used to remove a wide range of contaminants. Food protein production as biomass or normal food protein is dependent on the mushroom species and its ability to break down waste through the secretion of several oxidizing and hydrolyzing enzymes. White rot fungi linolytic enzymes have a broad substrate specificity, and they have been linked to the transformation and mineralization of organopollutants that are structurally similar to lignin. So, white rot fungal enzymes help in the breakdown of munitions waste, pesticides, polychlorinated biphenyls, polycyclic aromatic hydrocarbons, beach factory effluents, synthetic dyes, synthetic polymers, and wood preservatives. So, last is about microbial bioremediation. This bioremediation that is mediated by microbes offers a lot of potential for effectively restoring a contaminated environment. Recent research has focused on the process of bioremediation related enzymes such as oxidoreductase and hydrolases. A number of oil degrading bacteria make biosurfactants. The oil-water interfacial tension is reduced by these biosurfactants, which are typically of very low molecular weight. So, biosurfactants with a very high molecular weight operate as biodispersants by preventing oil drop coalescence in water. Heteropolysaccharides with active compounds such as lipids or proteins are very high molecular weight bioemulsifiers. So, in the last 30 years, degradation of polycyclic aromatic hydrocarbons has resulted in the isolation of various bacteria, fungus, and algae taxa or algae species capable of degrading low molecular weight pH. High molecular weight pH are generally resistant to microbial attack, and only a very few fungi and algae are capable of converting them or degrading them. So, xenobiotic degrading microorganisms have a significant potential for bioremediation, but their efficiency is dependent on the consequences of alterations that make them more effective and efficient at removing these substances. Chemotaxis is a key factor in improving pollutant biodegradation. So, conclusion. When a contaminant is normally present, the number of bacteria involved in the degrading process increases naturally, and when the contamination or contaminant is gone, the biodegradative population decreases. The treatments leftovers are normally basic compounds such as carbon dioxide, water, and microbial biomass. These are non-toxic. So, in many circumstances, it is utilized to completely destroy a wide range of pollutants. Many chemicals that are considered hazardous by law can be converted into less harmful or less toxic substances, thus removing the risk of future liability associated with the treatment and disposal of contaminants. So, thus we can conclude that bioremediation is less expensive, and it has a variety of other benefits over other waste removal techniques that are commonly used, and the type of organisms that we use for bioremediation depends on the nature of the hazardous wastes that are to be degraded. These are a few references used. Thank you.